Welcome to today's episode of iCANX Talks. My name is Christian Nijhuis. I'm from the University of Twente, and I'm a professor of hybrid materials for optoelectronics applications. And I'm very happy to have today Anja Boisen to give a lecture today. And you can see in this month's lineup, we have excellent speakers from all over the world, Europe, Australia, and of course also various countries in Asia. Today, I'm supported by three panelists, Liang Wang from Westlake University, Lingxiang Cheng from Beihang University, and Qingyu Jiang from Southern University of Science and Technology. But let me first introduce Anja Boisen from the Technical University of Denmark. She is heading the Department of Health and Technology, and also the DNRF and William Center of Excellence for Intelligent Drug Delivery and Sensing using microcontainers and nanomechanics. Anja is co-founder of several companies, including Kentian, Cellmeco, BlueSense Diagnostics, and LightNovo. She is also a member of several societies, including the Danish Academy for Technical Sciences, the Royal Danish Academy for Sciences, and she has countless of awards. But let me just highlight a few. What really stood out to me is that she is knighted by the Majesty of Queen of Denmark, and that she was also elected a Fellow of the Electrochemical Society. Today's talk is called Micro-Nano Engineering and Medical Devices. Anja, please take over the screen and deliver your talk. So I'll stop sharing so you can start sharing, okay?
Yes. Is it okay now?
See your slide.
Good.
Yeah.
Excellent. So thank you very much for the introduction, and I'm really happy to be here. And thanks to all of you who have organized this impressive platform that I learned and heard about from Alice. So today I'll give you two main stories on medical devices, but I'll sneak in a third one in the end, which is on in vitro systems. But before I do that, I'll just spend a few minutes on telling you where I come from and where we come from. So I'm from a center of excellence that we call Idun, and Idun is the name of one of our Nordic goddesses. So here you can see what we really strive to do, and that's bringing science to life. And I'll come back to that in a little while. But you can see we really want to do better science and to grow better scientists like you, and to also to create new businesses. And the way we like to behave, at least it's our aspiration, is we would like to be open, inclusive, to have ambitions, making a difference, and to collaborate. So when we were founded 9 years ago now, it happened kind of randomly. So we got two grants, major grants at the same time. One to continue what we've been doing for many years in nanosensors. So that's what you see on the right-hand side. And then a new grant to do something completely new to us at least, which is using micro devices for oral drug delivery. And I remember when I got those two grants at the same time, I was called to the office of one of the foundations, and I was told I cannot do this. I cannot administer two grants of this size at the same time. But then luckily they suggested, why don't you just put them together? And I would have to say what started as a really forced marriage is now a very happy marriage, because of course, at least to me now, I can see that there are a lot of inspiration for new applications when you start collaborating with people from the medical sciences and the pharmaceutical sciences. And to be honest, until then, a lot of the sensors we had developed, they were really searching for applications, and now we actually could get those ideas. Since then, we have been growing a bit. We have attracted additional funding, both to do some more basic research. This is our European Research Council grant, and something which is very applied, which I will actually show today, which is called the Bioinnovation Institute grant. To us, this motto, bringing science to life, It covers our ambition of implementing top-down micro and nanotechnology discoveries to important life science challenges. But it really also covers that we really strive to translate some of the stuff we do. And that's why we really like to do spin-outs. And you'll see some examples of that. I've tried to sneak it in the story that I have here. Some of the companies that we have established, you can see the logos here. And on top of that, we have some that are ongoing in the lab and that I believe will soon be spinning out as companies. And I will show you some of those today as well. This is us when we are all gathered. Maybe you can see that we come from many different nationalities. In fact, we are 25 different nationalities now. We also cover a wide span of scientific backgrounds, from computer science to pharmaceutical sciences, for example. And we collaborate with the University of Copenhagen and what's called Statens Serum Institute, where they do vaccine development. If you're interested, you can follow us on LinkedIn. This is also where we post news and open positions, etc. Where do we come from in terms of technology? And that's kind of our base, is very much micro and nano engineering. I actually did my own PhD in atomic force microscopy. So I started working when I got my first grant on nanomechanical sensors. We do centrifugal microfluidics. I'll show you that. Some of us really like to take things apart. So that's my colleague, Edwin. He loves to repurpose like a DVD player, for example. We build optical devices for Raman-based analysis, and then we do a lot of micro and nanostructuring, and you can see some examples of surfaces here that we are developing using, for example, our clean room fabrication facilities just next door to where I'm standing. But we also do a lot of fabrication outside the clean room in polymers, for example. So now to my two stories, major stories. So one is on therapeutic drug monitoring, which kind of covers also the story of two startups. And then there is one on the devices for oral drug delivery, which also kind of relates to two startups. So the first one is on therapeutic drug monitoring. And this kind of project started when we were approached by the head of the oncology unit at the research hospital in Copenhagen, Kjeld Schmigelhoff, and he is treating children with leukemia. When children with leukemia are treated in Europe and Denmark also, they get a very high dose of an anti-cancer drug called MTX. And the problem is it's really a very efficient treatment, but the children, because of the disease and also the size of the children, they metabolize these drugs very differently. So you have to monitor them closely to stay within the therapeutic window. Otherwise, you might end up giving too high a dose to a patient, and eventually this can be killing the patient. So the task is, when you do these treatments of leukemia, that you need to stay within the therapeutic window. So it means if you look on the curve to the left, you have on one, the x-axis, you have time. On the y-axis, you have concentration of a drug in the blood. And you need to stay within the gray area where the peak concentration in the blood is not toxic, and also the lower concentration is actually still having an effect. And to actually be able to provide this solution today at the Danish hospitals, you need to take samples, blood samples from these patients every second hour in the beginning of the treatment. And ideally, they would like to monitor these patients even more closely, so online, kind of. If you look at the analytical process of the blood samples from these children, you take the sample today, and in there you need to look for specific small drugs. You need to pre-treat the sample, and that's often done in a manual way at the hospital. And then it's sent for data analysis, and that's done at huge robotic facilities in a centralized lab at the hospital. So it's working in a hospital in Denmark, but there are still a lot of hospitals also in Europe who can actually not provide this treatment because they do not have the labs to run these tests. And on top of that, you would also ideally like to do it much more frequently than what's possible today. So that's why we then came in. We thought we had some ideas how to help Kjeld and all the doctors at the hospital. And we were also fortunate enough to actually get a grant from a new kind of foundation in Denmark. It's called the Bioinnovation Institute. and they support translational sciences. They're very much into incubating startups and to motivate scientists to kind of create new businesses. So we got a grant for 3 years, which I've never tried to get a grant like this, because it's research for 3 years, which should then end in a company. And the project has just finished here by the end of December last year. So here, first of all, you can see the team that has been hired in on this project. So to the left, you can see the very smart, hardworking postdocs that were hired in. But you can also see that there are other type of people on such a project. For example, I'll highlight 2 business developers, which I would normally not hire into a research project. So the core team, besides having different scientific backgrounds, also come from 6 different countries, which is bringing in a lot of diversity and new input in that way as well. The technology that we have been applying to detect MTX, which is a small molecule in a very complex blood sample, is Raman-based. So for many years now, more than 10 years, we've been working on something called surface-enhanced Raman scattering. So that's the cover you can see all the way to the left. I'll show you in a minute in more details. And we also actually know how to integrate this type of sensors into fluidic systems and to also integrate electronics together with it if needed. So we kind of thought that we had kind of a platform established to actually be able to do something in 3 years that could provide a kind of proof of concept for a little system to detect drug in blood samples. So before I go into some of the results, I'll just briefly introduce to you surface-enhanced Raman scattering. So Raman scattering is an old technique where you kind of send laser light at a certain wavelength onto a surface. And once in the light is inelastically scattered in the molecules on your surface or in your material you're studying, and the light comes back slightly shifted, so at a slightly different wavelength. And it's this shift in wavelengths of, what comes in and what comes out that's going to generate a chemical fingerprint of whatever you have in your material. It's nice because it's label-free and it doesn't require such a lot of instrumentation. However, the Raman scattering is very weak. So we would never be able just with Raman to detect small concentrations of drugs in a blood sample. We need to enhance the Raman signal, and we do that by using surface-enhanced Raman scattering. So it's been known since the 1970s that if you place molecules on a rough metallized surface, you can actually enhance the Raman scattering by 1 billion times. And that's actually how we entered the field initially, because we were interested in nanofabrication. So what we've been doing is to make rough metallized surfaces. And Michael was the first one here to do this. So he's been etching into silicon, and he can do that by a maskless reactive ion etching process. And then he's creating this nanograph everywhere on a 4 or 6 or 8 inch wafer. After you have created the nanograph, you can cover it with gold or silver, for example, and it's not a uniform coating as I've shown you here. It's much more rough. So these are the pillars when they have been etched. You can see you have nanographs everywhere, and it's covered with metal, primarily at the top of the pillars and then something in the bottom. And you can tune the height and the densities of these pillars. So initially, when we started testing the Raman signal from these substrates, we were surprised because they're really enhancing a lot, and also quite more than some of the competitors that we could find. And then Michael and some of our other colleagues started to investigate what's actually going on here. And we discovered that if you put kind of a liquid sample onto the substrate, and you can see all the pillars here, then when the liquid evaporates, the capillary forces are going to bring the nearest neighbors of the nanopillars together. So they kind of form small domes on the surface. And it kind of happens automatically. We need not to do anything. It's just happening. And if you look closer just at a few of these coated nanopillars, what's happening is that you can bring in an analyte. It could be the drug that we're looking for in a blood sample. And then afterwards, you lean your pillars together. And in this region where you have the red spot, this is called the electromagnetic hotspot. This is where you have isolated gold particles more or less touching each other, sandwiching the analyte you're searching for. And this is kind of a constellation that is going to enhance the Raman signal significantly. Other people are trying to achieve the same by bringing nanoparticles very close to each other. Here it kind of happens by itself. And indeed, they do lean these nanopillars. You can see that here. So this is just a scanning electron microscope image after we have been leaning the pillars and the substrate has been dried. When you have kind of etched these substrates, you can cut them into small pieces, 0.5 centimeter by 0.5 centimeter or bigger, and you store them in Eppendorf tubes. And then you can ship them around to collaborators, or you can use them in your own lab when you need to use them. So that was kind of the first spin-up that we had. It's a small company called medico, because then they etch these substrates with nanopillars on, and they sell them to researchers who would like to develop their own assays and their own protocols. So back to kind of being ready to integrate everything into a fluidic system. Prior to doing all this with the blood samples, we had been doing some work in liquid. Here you see a milk sample, and it's actually placed in a disc. So what you look at from the top is a CD disc with channels engraved, and in the middle, approximately, you have your source substrate embedded. It's diluted milk, and only half of the source substrate is being wetted by this diluted milk. Actually, the disc is rotating to work as a centrifugal platform, but you cannot see the disc is rotating because we illuminate with stroboscopic light, so we are only taking a photo per kind of rotation of the disc. But now it means that we can actually capture what's going on the surface as we are flowing in the diluted milk cycle. And maybe you can see, if the light is okay for you, that you have part of the source chip which is kind of dipped into your milk sample, but the liquid is traveling upwards. So it's kind of working, the nanopillars are working as a filter paper. It's dragging up part of the liquid, and all the small molecules will be allowed to travel upwards with it, whereas all the bigger proteins and so on will have to stay below because they're captured in the nanopillar filter. So it also means that you have a very clean region above, which kind of looks dry, but it's not completely dry. And below in the kind of submerged region, when you dry it out later on, you can see it's super contaminated. So if you do Raman measurements or source measurements in the top region, you'll see a very clean spectra of whatever you had in your milk sample. And here we had spiked it with a contaminant. And below, we couldn't see any clear signal because of all the other proteins landing on the substrate. So this was kind of our starting point for moving into blood sample analysis, that we knew that these nanopillar substrates, they also work as filters. So now to the blood story. So ideally, we would like to work with just a few drops from our patients. And before moving into integrating everything into a kind of a system, we started, this is Yaman here, he designed a little holder with 3 small source chips attached to it, and then below a tiny aquarium where he can load blood serum spiked with certain drugs. So here you see on top of the holder, you see the substrate, and then you receive serum, and you can mix in a little bit of methanol, and of course the MTX that you want to detect. The methanol will precipitate the proteins in your blood serum, and is also going to help to wet the source substrate. And then you're ready to do your measurements, dipping manually your source substrate half the way into your blood serum. The first data that Yaman then achieved showed that, yes, this is kind of working exactly like the milk example I showed you before. So the small molecules, the MTX, the drug, is actually able to travel up into the dry region of the source chip. And then we measure in that dry region, and we can actually detect the MTX in the blood serum. And we can also quantify it to a certain degree. So after establishing this first results and proof of concept, we actually started receiving the first patient samples from the hospital. So we got 2 samples from 2 patients at the hospital. So we got before treatment of the children, and then 24 hours and 48 hours after the treatment. And the before treatment blood samples we could use to establish calibration curves. The results you can see for one patient, we are kind of more or less on spot with what they could detect using their big ELISA robots at the hospital. For patient number 2, we are kind of 50% off. So it's not good enough. It was a first kind of try, very nice for that. And we also knew that we had too many issues. Until then, we were only using one peak in our Raman spectra, the MTX main peak, to identify what's going on. And the other thing is that we were just doing everything manually. So there is a huge risk that we are doing something, introducing errors. So that's what we started doing. First, we started looking at the entire Raman spectra, picking up several peaks in the spectra that we were interested in. And we did more advanced data treatment and machine learning. And I'm not going to go, I know it's a busy slide and the next one as well. But we were actually, by doing so, we are able to distinguish MTX from the compounds that it's being degraded into. So MTX and its metabolites, we can distinguish those. And that's something that you can normally not do with an ELISA assay. So it's already a competitive advantage with SIRS compared to ELISA. And we can also using this using several peaks in the spectra and using more advanced data treatment, we can actually distinguish MTX in a background of a lot of other drugs. And that's really important because these patients undergoing treatment for cancer, they are taking a lot of different drugs, sometimes up to 20 different drugs at the same time. Antifungal drugs, painkillers, etc. So that was about the data treatment part. Then there's also the automation part. And here we moved away from manual dipping into putting the SIRS chips onto a centrifugal platform. So here you see the entire CD disk. And in this case, you can do measurements on several samples at the same time. For each little kind of system on the disk, you have a reservoir where you can have your blood sample. You can do some pre-treatment, and then you have a place where you place the SIRS chip. All the fluidics will be moved around by spinning the disk by a simple spindle motor. And then it's the centrifugal forces that are generated that will actually move the liquid outwards. And you can do pumping, valving, etc. on this platform. So one of the first things that we implemented, and that's Laura's work here, was how to mix this methanol into your blood serum to kind of make that step more automated. And that has actually improved our kind of deviation quite a lot. So we have a much more reliable system by doing this on the disk. And the next step is then something we're still working on, but should soon be published, on actually integrating the sample pre-treatment of the blood sample also on a centrifugal platform. It can be simple filtration or solid phase extraction. Then in terms of instrumentation, Roman here has been building several prototypes. Here you can see two of the first ones in our labs. So these are portable devices that, if you look at the orange one to the right, below you see the disk with the SIRS chips, and you can spin it, all controlled by the software. And above in the hood, there's a spectrometer that can read out the signal from the individual SIRS chips that you have loaded in your system. So now the startup, because we did a lot of research, we're still doing research on this, but we had also kind of promised our sponsors that after 3.5 years, companies should spin out. And you can see the slides change a bit because these are some of the slides that have been used for pitching the idea for kind of raising capital. So this is from one of the first pitches that the team were doing. You can see the name is there now. There's a logo, Fluidlux Diagnostics. I show you a few of the slides just so you get an idea. We have decided not to incorporate yet because we got what's called a European Innovation Council grant, which will allow the team to work for 3 years on soft funding. So they will wait a bit, mature the idea before they incorporate as a company. Now I'll just share with you that we have done a lot of the initial work on cancer and the drug called MTX. But what's happening when you bring in business developers is that they're also going to look at the market size, and they will give you a lot of input on where you should go with your first product. So fortunately, you can say MTX leukemia, it's a small market. Very few children suffer from leukemia, and the market is therefore small. we actually also in Denmark sometimes have trouble getting enough blood samples to do the testing of our device. But there is another topic that needs a lot of therapeutic drug monitoring, and that's in the intensive care unit, especially for the patients suffering from sepsis. Because there, today, around 26% of these patients, they are overdosed with antibiotics. And it's because these patients are already very ill. So when you treat them with antibiotics, they already have organ failures, and you kind of have no clue how much of this drug is actually going to end up in the bloodstream. So 26% are overdosed, and this might actually kill the patients. On the other hand, 31% of them are underdosed, so they're not getting the treatment that they should have. So there's a huge potential and a big wish from the doctors in the intensive care to actually be able to monitor these patients online. And it has to be fast. That fits our device very nicely. And it also has to be at the bedside, which also is what our device can do. So this is kind of the first focus now for FluidLux diagnostics. Still, because all the initial data were done on MTX, they used the data from MTX to kind of show the proof of concept this is working. I'll just show the latest results. You can see that the source data from 3 different patients with leukemia is now very much in line with HPLC on the same blood samples or the immunoassay from the hospital robot. So it has really improved a lot and has come a long way, and now with a standard deviation of around 10%. So you want to follow FluidLux Diagnostics, there's a little bit of information here, and I'm sure more will come in the future. So that ends my first story. I'll now move on to devices for oral drug delivery. And here our kind of idea is that we would like to use micro and nano fabrication and micro nanostructures to make a little contribution to try to move away from all these needle injections. A lot of drugs today, they need to be injected because if you just swallow them as a tablet, they would be degraded in the stomach, or they would have no way to penetrate through the intestinal wall and into the bloodstream by themselves. They need help somehow, and devices, they can provide some of that help. Initially, when we started 10 years back or so, we were maybe a little naive. We were thinking, okay, we're going to make small microcontainers in polymers, and then we're going to load them with drugs. We're going to put pH-sensitive lids on top. And these microcontainers, a lot of them will align towards the epithelium, and then they're going to release drug in one direction. So you're not losing a lot of the drug. It's coming out very close to the epithelium. And then we have delivery. Here you can see some of these microcontainers. They have more or less the same size, and they also look a bit like small sugar grains. And since then, we have moved on to a lot of other devices depending on specific needs, and also because we actually face some challenges for some of our applications. But just to stay with the microcontainers so you can get an idea of how we're working. For our devices, we need to load them with drugs. And here you see an example where we have loaded microcontainers with powder drug. So that's kind of the more easy ways of doing the drug loading. And then you can coat them with pH-sensitive polymers, lids, we call it, on top. And these are normally polymers that you can buy commercially because they are normally used for coating bigger capsules. So this is for controlling a release depending on pH. So before moving on with the containers, I'll just make a little side story here, which also involves a startup, because we really wanted to see if we load drugs inside a polymer matrix in our containers, where do we have the drug and where do we have the polymer? And Oleksiy and Roman, they had built an instrument that can do 3D Raman mapping. And we used that to actually map out where do we have the drug in a microcontainer and where do we have polymers. And from that, actually, Oleksiy and Roman started using polarized light to do Raman imaging. And by doing so, if you look to the right top 2 pictures, you'll see that with normal Raman imaging, you'll just see the red part on that picture, which is showing you the drug, and the green part shows you the polymer on a tablet surface. But by using polarized light, which is then figure B to the right, you'll see that you can see a lot of more information. That's actually kind of giving you information about the crystal orientation of small domains on the tablet surface. And this is something that you would normally need to go to a synchronous facility to be able to do. And now you can do it with a, I mean, still pretty big, but just a Raman system in your own lab. So that started a company called Nano, and now they're also doing smaller instruments, one of them being a very tiny Raman spectrometer that they call Mini Raman, which was also recently published. So that was a little detour. Back to the containers. So if you really want to give these micro containers or similar small devices to animals or to humans, you put them in a bigger capsule so you know exactly where they are. So this is what you see here. And then you're ready to do experiments in rats, pigs, whatever. So some of the first tests we did was in rats, and we were using rather small molecule drugs just to see if this is working. So we worked with drugs like furosemide and ketoprofen. And this is an example here with furosemide, where we are putting small microcontainers with drug and a pH-sensitive coating on top into a big capsule. And then we're comparing it with the same amount of drug, but just in powder form and in one big capsule. And the blue curve shows what happens if you just give the drug to the rat in a big capsule. And the red curve shows if you're using the microcontainers. The peak concentration is more or less the same. But then if you look how long is this drug release going on, you'll see the red curve is just, I mean, we should actually have continued the experiment for even longer. We have a very prolonged release of drug compared to just using powder in one big capsule. And also, when we open up these rats afterwards, we could see that these microcontainers, they were actually embedded inside the mucus layer in the animals. So that's one of the reasons why we have such a prolonged release of drug. So we know that, okay, for some applications, the microcontainers, they're actually working. So then we moved on. We've tried a lot of different things to deliver probiotics, antibiotics, insulin vaccines, and we've also worked a bit with gut sampling, which is kind of a slightly different topic. So in some cases, this works very nicely, for example, in the treatment of bacterial biofilms. But there are other applications where we really struggled. And I'll share one story with you where we struggled a lot. So in Denmark, we have a company called Nano. They produce insulin used for diabetic treatment. And we kind of felt, okay, we need to kind of make a try at doing oral delivery of insulin. Normally, you need to inject the insulin. However, this is a difficult task because insulin doesn't, I mean, even if you can protect it all the way to the epithelium, it will not go through the cell layer. It needs help. And one of the ways you can help is by using something called a permeation enhancer. This is a chemical enhancing effect that will actually go in and open the tight junctions between the cells in the cell wall in your intestine, and then the insulin will be able to go through and into the bloodstream. So we thought, okay, we'll just mix the permeation enhancer, C10, and insulin into these small microcontainers, and then we'll see if it works in rats. Before moving into animal, we did some initial tests just on cell layers mimicking the intestinal wall. So this is Jakob here, who did these experiments in collaboration with University of Copenhagen. And he has a layer of cells, cargo-2 cells they're called, and on top he places an array of these microcontainers filled with permeation enhancer and insulin. And what he could see was actually that you can get insulin across these cells if you put the microcontainers very close to the cells. If you lift them 0.5 millimeter away, or 1 millimeter, 0.2, 0.5 millimeters away, already you have very little release of insulin. So it kind of told us that we had to be super close to the intestinal wall to have any hope of any release of insulin into the body. We tried, however, in rats, and it didn't work. So we had to think differently, and this brought us to this, as we call it, the self-unfolding foil. So what you see to the left is some foils that have a lot of cavities in them, to hold drug, insulin, for example. And it's an elastomer. It's called PDMS, this material. It's flat, and then you load it with the drug and coat it, and you roll it up into a capsule. And when the capsule dissolves in the intestine, as you can see to the right, the foil really would like to regain its original shape. So it's going to press against the epithelium, and it looks very uncomfortable here in this piece of tissue, but it's because there are no organs around. But you can clearly see maybe some of the small structures, the cavities, through the tissue. So now we are very, very close to the epithelium. So after that, we moved into animals, and here is the first proof of concept from 2020, where we could for the first time see just a tiny amount of insulin in the blood of the rats that we were studying. So we thought something is actually working here. Now we have optimized the device, so we have much higher availability of insulin. Now it's 2-3% that is actually coming into the body. And if you do the same in pigs, and here we're using not insulin, but a small peptide called nisin, which is having antibacterial effects, we actually can improve the oral bioavailability by something like 300%. So, I mean, this is something that for us is very, very promising and something we'll continue to develop. What we are trying now is to embed some of these devices like the microcontainers or the foils with small devices or robots that can swim by themselves. Because this can be a way to travel the last distance that we might need to penetrate the last piece of mucus in the intestine and getting very close to the intestinal wall. So Gianna was the first one to bring in micromotors in our lab. So the first study she did was to kind of take magnesium microparticles and coat them with a drug, and again coat them with a protective pH-sensitive layer. And then you can make them swim in a solution, because the magnesium, which is exposed on the kind of bottom side of these particles, will react with liquid, and the bubble generation that comes from that will propel these small motors forward. And after loading them into containers, as you can see in some of these top-down images of the containers, we could actually see that, yes, indeed, the micromotors, they come out and they swim kind of straight in the mucus because of the viscosity of the mucus. And interesting also, the microcontainers can only leave in one direction, so we have more or less secured a unidirectional release of the drugs that we load into our microcontainers or our foils. Another thing that we are really struggling is for some applications, like, for example, vaccine delivery, we want to make our devices stay for longer in the gut. Especially in smaller animals, they are cleared out in just a few hours. So we need somehow to increase the retention time. And one of the ideas, because we are very much in love with nano and microengineering, is to embed structures on the surface of some of these devices to have some mechanical effect. So Lucas here, he did some 3D printed structures, kind of copying also like bacteriophages designs. And in vitro studies, they really see that, you know, there is a difference. And like the phage-inspired design has an increased retention, but it's like a 10% increase. It's not, you know, 300%. We really needed to go down in size of these devices. So we started building our own 3D printer. So here you see Tianjin, and it's also part of Edwin's work. So they hacked a Blu-ray player, and they took out the blue laser from the Blu-ray player to use it instead for cross-linking a polymer. So the Blu-ray player is cool. It has a lot of technology inside it. It's kind of disrupted already, but the technology is fantastic. So the spot size of the blue laser is less than 1 micron. And you can control the laser. So you can actually use it to, with very fine resolution, to cross-link a polymer and build structures. Here you see some of the initial structures, but it has then improved a lot. So now we can cross-link not only a polymer, but we can also mix contrast agents into the polymers so that we with CT, X-ray, can actually track our small devices in animals. So here is one of the examples where we are printing more advanced structures using tangential printer, and then we have been testing it in rats to see that there is some difference between the different designs, but certainly something that needs to be improved even further for the future. Again, a little startup story to go here, because the printer itself that we've been using to make our medical devices is kind of a cool technology. Provide you with a cheap solution for doing very high resolution 3D printing. So the team here, they can do all sorts of structures like hollow needles for injections through the skin. They can also do more fun stuff like the Eiffel Tower, or here, as you can see, the Notre Dame Cathedral, and they're very proud of all the fine details and the overhang structures here. So if you're interested, I'm sure that they are very interested in future collaborations. This will move me to kind of a little other application involving capsules and microcontainers. And that is in kind of sampling, what do we have inside our gut? What type of bacteria do we have? It's becoming a very interesting topic to actually know more about our microbiome. And it's a challenge actually to take samples from all of the intestine. You can put in devices, but they cannot go all the way through, like an endoscope, for example, and it's not very comfortable. So we would like to also make some contributions here to make capsules, devices that can take samples from the gut, and then they come out and we can collect whatever they collected. So the first results we did here was when Savesh and Fatima joined us. So Savesh came from a background with micromotors and Fatima with tissue engineering. So they actually repurposed some of our microcontainers, as you can see. They loaded them with propellants that could propel the microcontainer a bit forward, but also with reagents that would grow a hydrogel tail. So as the container is moving forward, it's growing a tail that is going to encapsulate whatever it meets on its way. So they could show that they could encapsulate some cells. And that was kind of really exciting to see. Of course, the next was then to take some of these micro containers with a gel-forming tail and give it to a rat. And then afterwards, try to relocate and find some of these containers when they came out of the rat again, and to analyze what have they actually collected inside the gut of the rat. And if we then could compare what bacteria do we have, if we open up the wrap and look in the intestine, what do they have of colonies in there, and compare it to what we had collected on our micro device, we could actually see a very nice agreement. So this was kind of our first step into gut sampling. What we now do is more big devices, because they're also much easier to collect afterwards when you need to harvest the devices and analyze what's inside. Here you see some of our first prototypes for doing these type of experiments, but in pigs. And I think that soon we'll have our first paper out on sampling from the stomach in big animals. So that was kind of the two stories, main stories that I have for you. I had one on therapeutic drug monitoring involving medico and Fluidlux diagnostics, and one on all drug delivery, which I tried also to integrate two stories, one on Novo and one on Atto 3D, which is now called Micro 3D. I have a little bonus story, which is the project called Cell Loop. So here again, we are working with centrifugal microfluidics, but now we are using this steady flow of liquid to grow cells, human cells or bacteria. The motivation, or kind of the idea, is that a lot of drugs are being developed, a lot of the drugs are failing, and they need a lot of preclinical studies, and they also need a lot of animal testing. It has now been more or less approved, or it has been approved by FDA, that it's not longer required to do a lot of animal tests before going into human trials. So a lot of companies and also research groups are now focusing on growing these different organs on a chip to kind of help actually testing drugs in a much easier way without involving a lot of animals. So we thought, okay, here we have a platform. We have these rotating discs where we can integrate growth chambers for our cells. And we have a waste chamber around to collect whatever flow or buffer comes out. So the idea is we want to grow all cells in flow because this is mimicking much better what's going on in the body. And using a rotating disc is a very gentle way of doing it. You need no external pumps. You don't have issues with any bubbles. And you have a very steady flow. And on top of that, it's a tiny device and you can put it anywhere in an incubator. So this is the idea. This is what we're doing. And here you can just see some videos of what it looks like. You have all of these spinning discs. And if you want to have more cell growth chambers, you just stack the discs on top of each other and they all rotate together in an incubator. Again, it's the spinning speeds that's going to control the liquid flow over your cells. So we have been growing cargo-2 cells that are mimicking the cells in your intestine. And when they grow in flow, they become much more natural or like your intestine, because now they start forming these villi structures that you also have in your body. And on top of that, the cells also connect much better. They become tight. And this is what we can see when we then try to test these cells in our platform. If we try to, detect any amount of drug going through these cell layers, if we grow these cells just in a static culture, kind of like in a petri dish, and see how much go through, it's not tight enough. But if we do it on our culture in perfusion, we get a much tighter layer, something that's much more resembling what you have in your intestine. So here they are also looking into kind of, can this be translated? Being a business model where you buy a little motor and all the discs and holders for doing your experiments, and the chip itself, the organ chip, is what you consume as an end user. So the benefits is that it's easy to use, it's high throughput, and it's also a very small size. And it also has some advantages in terms of what you can grow on top of it. And here is the team that is pushing this forward. So I think this will bring me to the end. I would like to, in the end here, thank all of you for listening. I would like to thank the sponsors that have been making all of this possible. And also at the end here, really thanks to all these amazing people who are making it so much fun to come to work every day. So I think that's the end. Should I stop sharing?
Yeah, thank you very much for a beautiful, inspiring talk. Yes, you can stop sharing. So then I can start again. Yes. No, so thanks again. And now let me introduce our panelists. Today we have Liao Wang from Westlake University as one of the panel members. He is currently an assistant professor. And he received his doctorate degree from the Technical University of Ilmenau in Germany. And I just found out that he actually spent some significant time in Münster as well, which is very close to where I am, from 2017 to 2019. He worked as a postdoctoral researcher at the University of Connecticut, and he is specialized in aluminium-based 3D lithography to fabricate structures and study their structure-activity relationships in optoelectronic devices for biosensing. He published in all important material science journals, including Nature Nanotechnology, ACS Nano, of course, and Advanced Materials. And he also has won several important fellowships, including the China National Scholar for Outstanding Cell Finance International Students. Now let me move on to our second panelist, Lingxiang Zhang from Beihang University. He is there a professor since 2019. He obtained his PhD degree from Ohio State University in 2016, and then joined the University of North Texas as an assistant professor. His research is supported by the Xiangjiang Scholarship and the National Young Thousand Talent Program, and he is covering cell diagnosis and therapy, biochip, and nanoelectroportation. He has authored more than 100 papers in also excellent journals and won many awards. But maybe that is also really very good in this context is that he also started a company called Cellucure Biotech that has currently a market value of 50 million. And then our last panelist for today is Zhengyu Jiang from Southern University of Science and Technology. He received his bachelor from the University of Chicago and his PhD from Harvard University. And he began his independent research career at the National Center for Nanoscience and Technology back in 2005. In 2018, he was appointed chair professor at Southern University of Science and Technology, Shenzhen, China. He has been awarded a lot of grants, won many awards, but I think it is quite, he's really established, having published more than 400 papers, and also incorporated already several companies. So let me then move on and let's start our panel discussion. And maybe we can start our panel discussion in the same order as I introduced them. So I would like to ask Liu Yang to start the discussion.
Oh, okay, thanks, Chris. And thanks, Anja, for your excellent talk. So it's great honor to be here to join this discussion. So maybe I can start the question regarding the SARS, because actually I also did SARS quite, as I mentioned, I really very good behavior in advanced materials a long time ago. And so because I am doing micro nano applications, I also want to use it for SARS substrate. You know, the SARS are being used for biosensors very, very long time ago.
Yeah.
But commercialization is not very good, you know. Because I know you established a company to sell the SARS substrate based on your silicon etchings. And but I have quite a few questions regarding this, you know, SARS substrate. And the first one is because, you know, in the SARS is based on the hotspot. And if you want to get a uniform signal, you want to make sure this hotspot to be very uniform if you want to capture signals from the random spectra. So how you think, the first question, how you think this randomly self-sampling etching, did you think it can give you very highly repeatable and robust search signals if you are doing some molecular sensing? My first question is, thank you.
Thank you, and thank you. It's a really excellent question, and something that we have also been discussing ourselves. And we have also been looking into if we should really go into also making, you know, much more structured surfaces using certain types of lithography, for example. We're staying with the nanopillars for now, which is a kind of a very stochastic process, as you say. So it looks a bit random, and it is also, but the density is kind of uniform. And when we measure, we are never just measuring in one side, and the spot size has certainly, of course, also a certain area. And then we are mapping, you know, maybe a huge area of the chip, and then we're collecting all the data. And I think this is why it goes well. So we have a surface which is enhancing a lot, and we are collecting from a very big area where we are then doing the averaging. And that's why we can then get, I would say, very low standard deviation now, and even doing the concentration detection of the MTX. I have to say that it's really important that you know what's going on your surface, so that if you have background contamination, that's also something we have been dealing with. You need to kind of look into where is this coming from? How do I store my SARS substrates, for example? Or is there any other way when you do the data treatment afterwards that you can correct from whatever data or every background you have? But I would say how to keep your SARS substrates clean is an important part of it.
Okay, thank you. So I have my following questions, because, you know, when you mirror SARS, you normally, as you mentioned, but you will put your substrate in this solutions contain some biomolecules. You're waiting for this molecule to dry and attach to the substrate, right? So then they have, you know, for the most of these solutions, when doing the drying, they have covering effect, right? So the concentrations for small molecules will be not uniform across the substrate. So how do you to guarantee you, when you choosing different regions, they have uniform search signal? So how you can tell this is concentration. As you mentioned about it, you are comparing your result with this HPLC. But
Yes.
how do you think you are, this cross, you mentioned about your surface is around 0.5 centimeters squares. So when you're doing the drying, so actually the different spots, do they have a really uniform concentration, or you need to do some calculations? So how do you think this is possible to comparing with HPLC? Thank you. Yeah.
Yes. So I think that one of the solutions for achieving this, so that we can actually compare with the HPLC, is that we do some sample pre-treatment of the blood sample first of all, and then that it's integrated in this centrifugal microfluidic platform. So the liquid handling is the same every time, and then that we're doing this, that we're only dipping half of the surge chip into the serum. And then we are letting, so it's about how you are wetting the substrate. So it's kind of traveling upwards, and you kind of can see that there's a band where we have the surge signal. So we are kind of using that the MTX we're looking for is a small molecule. It will be traveling up with a little bit of the liquid onto what we call more the dry region of the surge substrate. And that makes the results much more reliable. We remove a lot of the coating, as you say, is not happening on the surge substrate.
Oh, okay. Thank you very much. So maybe I have one more question.
Sure, continue.
One more question about the search part, because for most of the applications they want using search technology, they will be put it they know they really known biomolecules, but when you test the really samples, they have lots of unknown molecules mixed, right? So
Yes.
you need to. But the problem, for most of the molecules, maybe they have search patterning, the same search patterning. How you think the technologies can help to identify different molecules? They have similar search patterning, and this is very because when we are reading not our manuscript papers, they always put the known molecules. But in real sampling, you mentioned about in real sampling, you need to put a lot of unknown stuff inside, right?
Yeah.
How you can make it work. So I saw you using some microfluidic to separate the different size of molecules.
Yes.
But for the same size, how you can make it work. So that's very, very amazing. So I think it's very important if you want to really to make the search can be commercialized. Right? I think that is an important point.
It is important. Yes, it's a good question. I can say that we are receiving patient samples from the hospital, and these patients are receiving a lot of different drugs. And it has not been a problem to pick out the MTX signal, for example, from those patient samples, even though they get maybe 20 different medications at the same time. And I think it has to do with we know what we are looking for. We know exactly the spectrum of what we're looking for. And we're not looking just for one peak, as some people do. We're trying to look at several peaks or the entire spectrum. And then, also you can see more and more people are going into machine learning as a way to kind of identify and learn and pick out the signal. So I think it's a combination of making everything as automated as possible, as reproducible as possible, and then also the data treatment afterwards. Then it is actually possible.
Oh, so with this deep learning, so make it possible to identify. But before, without the deep learning, it looks like it's very difficult, right? So you need to figure out which is which with the same Raman.
And I think also if you're just only looking at one peak in the entire spectrum, it's going to be difficult. But there's a huge risk that something else is going to be right on that peak. And of course, you're reducing that risk if you're working with the entire spectrum.
Okay. Thank you. So I'm all set. So maybe I just hand over to Lin-Chen. So thank you, Anja. Thanks for a lot for you.
You're welcome.
Yeah, thank you. And of course, this is a panel discussion, so other panelist members can always jump in if they would like to. And I would also like to reach out to the audience. If you have a question, please put it in the chat box, and then later I can perhaps read it out to Anja. So let's continue now the panel discussion with Lingshan.
All right. Thanks for the introduction, Christian, and very nice talk from Anja. Thanks so much for the really nice talk, and I was really impressed about this presentation because it's covering a lot of things. It's very informative, ranging from micro nano devices for sensors and also the drug delivery for therapy. Actually, I was also working on something in drug delivery, but my research is a little bit different. We are working on some local delivery. You are working on some micro containers to do the go through the circulation or something. So due to I have a lot of detailed questions to ask, but due to the time limitations, I think there are two more important questions that I'm more interested. The first question is regarding the what is called a permittivity mucus permeability enhancer.
Yes.
So I see very acute micro containers and which contain some drugs inside, and it's patch on the mucus, right? Or
you can say
like
so,
nearby
yes.
the mucus, and then
Yes.
release some drugs to increase the permittivity of that mucus site, right?
Yes.
So maybe I lost one detail. So what is permittivity? What is a drug for the permittivity enhancer? Is that small molecule drugs or something inside?
Yes, it is a small molecule. So these are something that are also commercially used in a few products. I think it's still very controversial to use them because there is a risk. I mean, you are kind of locally damaging the tissue for a while. And you can also see that when you work with cell layers. So when we used, there's one commercial or one molecule called C10, there's another one called SNAC. And if you put them in the containers and very close to your cargo 2 cells, you'll see that you are really damaging the cells if the concentration is too high. And then you just burn a big hole and then everything is going through. And that was not really the intention. So you really have to lower the dose and be careful and understand what's going on. So we are also looking, I mean, maybe you're familiar with some of the work from Bob Langer's lab at MIT, where they do more, the injections all the way through the epithelium, which of course also might have some concerns. But we are also trying to explore other means, something in between, not using the chemical enhancers, but that was where we started.
I see. One thing to discuss further is because I'm working on this as well, because we are also making a devices which is actually a physical devices. It's using electrical field to increase the permittivity of a mucus or like the epithelial or dermis. So to me, we are also comfortable with its efficacy to to make the skin or mucus like more permeable. So to discuss with you about these things, because in my opinion, physical, for example, the electrical field is still cannot like 100% to avoid the damage, because you cannot control the electrical field very well, but its efficiency is also it's also powerful. So is there any like a combinatorial design principle to to combining the physical and also the chemical together, make like reduce to reduce the damage to the cells while significantly increasing the efficiency, also increase the the distance for the drug delivery?
Yes, I think that would maybe be a way to explore more, to do a combination of technologies to see. But I also really like what you're working on. That's something we've also been discussing about applying an electrical field, because it works a lot on the skin. There has been a few also in the gut. So yes, I think that we should explore a lot of different options because this is not easy, and we need more people to look into this for sure.
Yeah. All right. Thank you. The second question is actually it's not a detailed question for the experiment. Actually, what I'm asking is the philosophy, because I see your research, and you actually shared with us several different experiment or different stories ran from from the spin disk microfluidics, and also the thirst for the molecular detection, and also the drug delivery, and last several slides covering the organ on the chip. Is there a main lead, like a logical lead to organize you and the group members to to work from one step to step, or like you just push all the field in parallel? What is your working principle?
No, I mean, it has evolved over the years. So we started only doing the sensors. And then we got 9 years ago, we got this grant to also do delivery, but we were using all the inspiration from micro and nano fabrication that we had been using in the sensing part. And then we really had been working with centrifugal microfluidics, and that's as part of our sensing activities. So whenever we could see something that we could put on a disk, we tried to do it because it was there already. And I think that's how it evolved. Kind of the overall umbrella now is that we would like to work on kind of this dream of swallowing your own doctor, so that you can both sense and sample and deliver in the gut. And in order to do that, because this will kind of bridge everything we're doing more or less into one goal. And in order to be able to do all of that, we also need some energy in the gut to help us, and we don't want to swallow a battery. So we have a new grant now where we are looking into doing energy harvesting inside the gut. So either using somehow whatever we can use in the stomach or in the intestine to generate the energy that we then will use for sensing or for helping us to get the molecules across the epithelium. So this is kind of now unifying everything we do. I'm very happy for that.
I see.
So what is the generation mechanism? You use the pillars that you talked about in the beginning to generate the electricity?
So we're exploring a lot of different options. You can also find a few other groups are looking into harvesting the acidity in the stomach can be used to make a battery. You can use the peristaltic movement in the intestine to harvest that. You can make microbiofuel cells using the environment or bringing things down that are not toxic. So we are exploring many different possibilities here. It's kind of a new field, at least to us, and in general, a rather new field, I would say.
Thank you.
Welcome.
Is a motivation to to drive you and your group to do some like micro nano devices for medical field? I mean, it's because technical oriented or medical oriented. For example, you find some papers, or you see the, like for example, the microfluidic devices in the lab may be useful for some medical devices, or you talk with some medical doctors from like each specific department in the hospital, and they think maybe this device is useful for their specific questions. Like which one is the better way you want to move into that field?
I think we do both. And sometimes it's just pure serendipity. So for the cancer detection or the drug detection for cancer treatment, it was by a discussion with the medical doctors. And I would say talking to people who are helping children with leukemia is it's very motivating. You just want to help as much as you can. And this has been motivating for all of us to drive this as much as we can. And I would say the medical doctors, and I think that's nowhere where you go, they are very passionate. They really want to help their patients. They will invite you in. You even meet patients. You can talk to parents. So it's super motivating to see you can actually do something to help. In other cases, I mean, for how we started growing cells on disc, it was actually we were doing microcontainers to do delivery of antibiotics. And we had to do in vitro testing with the group at DTU who are really famous and good at doing bacterial biofilms. And then my PhD students, they went down to the lab to do the in vitro studies, and they saw, I didn't show it in my slide, but it's just a lot of small microfluidic chips, a lot of tubes everywhere, bubble trap, huge pumps, several liters of growth medium because there was a lot of waste in all the tubes and so on. And then they kind of came back and said, I think we can do this on a disc instead. So they were really motivated by just that it was so complex. It was not easy to operate, and they would always get stuck with the bubbles. So I think that it's another way of getting the inspiration and the motivation. But of course, you need to know that you have something at home in your own lab that might be able to do this in a smarter way.
All right, thank you so much.
So may I follow your question? So because you mentioned about beside this source technology, there are other important optical biosensor technology. We call it surface plasma proton based optical sensor. So actually, it looks like the SPR they have commercialized the most successful than such technology. Even they are developed the similar times. So how about your opinion about the two technology? You mean which ones have better features for small molecules like identification like this?
I think time will show. I think that the reason why SIRS is so hard is because you need to somehow have a substrate or a solution with nanoparticles that's behaving the same way each time. And then the integration into a fluidic system. I think that's probably why it's taking a lot of time to do this. I think the benefit is it's a completely, I mean, you can do multiple detection very easily. I mean, you could pick up signals from many drugs, different drugs at the same time using SIRS. You can distinguish, in the same blood sample, many different drugs. It's a spectroscopic technique. It's label-free. I think that can be playing to its advantage. But I guess in the end, we're going to see. And it might not even be the best technology. It's going to be dependent on so many other things in the end when you go into business.
So perhaps we can move on with the discussion to Xingju, because also Alice joined and also would like to give her a few minutes later. But let's continue with Xingju first.
Okay, thank you, Christian. And thank you, Anja, for giving really a very impressive talk on microfluidics and nanotechnology for diagnostics and drug delivery. So I have a question. My first question is about the the analytical part where you use the SIRS to detect methotrexate. So I guess somebody was asking you about the comparison with SPR, but I'm more concerned about the comparison with immunoassays. And so I think by far the most successful commercially sort of non-amplifiable assay, I guess. So you exclude the nucleic acid. The most successful commercial assays are mostly both based on immunoassays. So I was wondering if you have compared your SIRS-based assay with the I guess competitive immunoassays for methotrexate. Now I haven't checked if there are any commercially available, but I'm pretty sure there are ones available, at least in the research labs. Have you checked in terms of the robustness and the analytical performance in general, sort of the comparing SIRS to the immunoassay-based detection for MTX?
Yes, I mean that we do in collaboration with the hospital. So they are doing the immunoassay and the HPLC testing for us most of the time. Sometimes we do the HPLC here as well. So yes, I mean it's very comparable. I would say this is a technique, and we didn't know that from the beginning, but our technique can actually distinguish the different metabolites, and the immunoassay cannot do that. So that's giving a false reading. So you will both get the reading of the MTX concentration, but also the concentration of some of its metabolites. So this is, I mean, that could be a selling point for SIRS. And then the SIRS here, but you could of course argue that there could be immuno-based assays that could be as small and fast as ours, but we have kind of, it's completely label-free. So within 10 minutes, you have the results on the patient samples, including all the pre-treatment and everything.
Thank you. So my second question on the analytical part is, so you talked about MTX and antibiotics.
Yes.
So I was wondering if you have looked into other medical applications in terms of detecting small molecules within the blood. For example, some hormones vary quite dramatically over the course of the day, and that can be important for detection of some diseases. Would
Yes.
this SIRS-based method be applicable for those as well?
I think so, and thanks for the suggestion. And what we have been doing, because we've been working alongside business developers, is that we actually got these different inputs on what would be important to concentrate on, and those were based on quite a lot of interviews with medical doctors and practitioners. So what we have been looking into and also focusing on is epilepsy. Is a field where I mean, when you are diagnosed and when they are still kind of, it sounds like experimenting a bit on what drugs will work for you, you need to be monitored very closely. And there is a need there for actually doing, you know, a fast screening of what is the concentration in the blood of these anti-epileptic drugs in the patients. And another field is for mental diseases, is also a field in need of more monitoring of the patients. So, but there are many. So it's been kind of based on a lot of numbers that we have zoomed in now on antibiotics and intensive care unit. But there are certainly other fields where this is needed. I think maybe my
still there, or you look frozen?
He looks frozen.
Should be
should be
frozen.
He stopped. Well, then I'll take the opportunity in the meanwhile, hopefully, since you can reconnect, to give the word to Alice, because I think she has a different type of questions for you already.
Okay. Oh yes, and yeah, I'm so excited to hear your talk. You know, 1 year. I mean, last year this time we met in Switzerland. Remember, yes. APFL? We talk about this. Invited you to give a talk. You know, yeah, time fly, but so exciting. Your talk was so nice. Yeah, even give me more thinking about that. And congratulations. You know, not only for your research, also for move to the industry. You know, to the market. I think the startup is so nice. I think we are right tuning for, we want to have a Davos summit this year is try to link, science, technology also with entrepreneurs. Yeah. Yes. So yeah, you already did that, and I realize you recently win some honor, right? Some awards, super nice. Yeah, and leading the innovation centers. So really proud of you. And my question is, how you handle, such a wonderful life, a beautiful research. I know you have a very lovely family and now so successful, not only in research, also in the, moving forward. So how you handle this? How you balance that? Okay, please.
That's a good question. I think, when I was younger, I was also very stressed. So I learned, you need to also prioritize to sleep and to eat and to be with the family you care about. I think these are important things as well. Then in the end, you get more energy and you can actually, you're capable of doing more. So that would be maybe my first advice. be sure that you sleep enough and you eat enough and you exercise, and you are also with the people that really are important to you. And then, of course, enjoy the work that you are doing. And I would say also, at least for me, it's important that I'm in an environment where there is a trustful relationship and where I'm happy to go to work every day. And I'm very fortunate now. I'm kind of in a situation, and I have the possibility to kind of create the environment, or at least, you know, set the framework for that. But I really believe that there should be trust at the workspace, and that people, that can be healthy collaboration. But I think, I mean, unhealthy competition can hurt a lot. So I would think about where I am and what the environment I'm placed in when I'm working, especially if we're working very long hours. You have to kind of think and consider, is this where I want to be for many years?
So
Okay,
sorry,
how
I was
nice.
disconnected just
Yeah,
now. Can I still continue the question, or am I not
Yes,
allowed
yes,
to?
please.
Okay,
Yeah,
sorry.
please.
So my last question actually is actually a short one. So I can't help noticing that you were founded by Novo Nordisk,
Yes.
who has become the world's largest pharmaceutical company by selling semaglutide. And you didn't mention anything about semaglutide. Why is that?
No, yeah, it's actually because there is an arm's length between the company and the foundation. So the foundation, you know, when they started giving out money, it was a very narrow topic within metabolic diseases, and now it's a more wealthy organization. I should say it's probably the largest foundation in the world now. So they have also broadened out a lot. So they do research, or they sponsor research in many different fields. Also kind of in something that's close to engineering, as long as it has some sort of health application or planetary health. They're also interested in that. So you don't need to do anything with insulin or semaglutide to get money from them.
Great, thank you. That was really wonderful. Thank you very much.
You're welcome.
Yeah, so let's thank you. So let's go back to Alice again. Right, Alice?
Oh, yes. I was just following questions about, you know, also Xinyu mentioned that. No, Anja, you like amazing. You have a super team. The last page, you know, show that looks like, you know, at least 50 or 60 people, you know, different colors, I mean, different cultures working in the same group. You also talk to so many different kind of parties, right? Investment and also students, hospitals and the patients. So can you tell us your trick how to communicate? You know, how to get, you know, communicated with different people.
Oh, communication is difficult. I think this is, you know, when things go wrong, it's lack of communication, or it's lack of alignment of expectations. So I think that, yeah, it is not easy. And if you have 25 different nationalities, there's a pretty high risk that you misunderstand each other once in a while. And that's actually, I think it helps that we want to improve this, and that before we know that it can be a challenge. And we actually also got a grant from another foundation called the Lundbeck Foundation on diversity. So we have been hiring in a person who is very good in cultural intelligence, and he's been testing all of us and our cultural intelligence before the project started. And then he's been here observing us, interviewing us. And now we have done a second test, and we have also been asked to do written feedback to some of our colleagues. It's anonymous. And then we received training on cultural intelligence. And, you know, from Denmark, we have a very flat hierarchy. And when I grew up, I was given a lot of freedom, and I hate when people tell me what to do. So I would naturally, when I should supervise a PhD student or postdoc, I would say, I think we should move. I would like us to achieve this and this, but I would not make a very detailed plan. But maybe if you grew up in a different culture, you expect that I'm the one telling all the details. And it took me a while to figure out that I cannot expect that everyone wishes the same as me. I need to really also adjust to, you know, the person and where you're coming from and your cultural background. And I think that's not easy, because if I'm tired, I just, you know, fall back in my ground state, and I'm just, you know, so it really takes extra energy to have this more awareness of that we are not the same, and our background is not the same. We were not trained in the same way. We didn't grow up in the same way. And even words can be misunderstood. The way we use our hands, or, you know, so many things can go wrong. But at least if you are aware, it's the first step.
Okay, I think respect, right? To try to understand, yeah, different cultures, respect the
Yes.
different kind of, you know,
Yes.
personalities, and
Yes.
this
Yes.
was a keywords. Yeah. So I'm so proud to see that in your team, especially your, you know, startup team, you have a Chinese one, right? Yeah. So yeah, as a young guy, it looks so nice. We're proud of that. You know, recently we really think we got some wrong message, you know. Looks like they don't welcome Chinese. I see in your team, Chinese is really welcome, right?
Oh, yes, 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 yes. And I also just, in terms of diversity, I really believe that diversity is source of inspiration and innovation. It might be that there will be more frictions once in a while, but it's also so much inspiring, and it's so much more creative when you bring in people with different perspectives. I think we should all try to be more diverse. So
Yes, iCANX is diverse, right, Chris? Am I right?
Fantastic.
Okay.
Yeah, absolutely fantastic. Yeah, I couldn't share more. I think that is one of the privileges of definitely being of this part of Europe. I think the Northern European countries, I think, are really progressive in that sense, and the Netherlands in a sense too. So it's really fantastic to see such a wonderful example of it today. Yeah, thanks for sharing your experiences with us. Yeah, so anybody else has still some questions, or? Because otherwise I would like to take the opportunity to thank Anja one more time. And then I think we can hand over the fantastic, yeah. Oh, we already go to the iCANX, sorry, the award slide, of course. So this will be sent to you later via email.
Yes.
So thank you again for not only the talk, but also the wonderful discussion we had. Yeah, then I would like to advertise one more time the Davos, the real life summit meeting from iCANX, which will take place on the 11th and 12th July. We have a wonderful program, and the latest I heard, already more than 500 people have registered for the event, so it's going to be quite big, spanning the entire range from science, innovation, technology, management, entrepreneurship. So I think it's going to be a wonderful meeting. And then a slight advertisement for next week's contribution that will be from InQ Park, from Keist, and he will be talking about smart environmental monitoring by low-power, self-powered gas sensors and artificial intelligence. So that pretty much concludes it for today. So thank you to you all, and also the audience, and hope to see you all next week again. Bye-bye.
Hello, ladies and gentlemen, my dear friends. Welcome to iCANX Davos Summit. So this year we're going to have this big event in Davos, Switzerland. We have 6 topics: innovation, science, technology, investment, management, and entrepreneurship. Short for It's Time. So it's time for us to go together. It's time for us to all these inspiring talks for the future. We'll have Nobel laureate forums. We'll have president of academic firms. We'll have president of university firms, all these big names will be here to talk about the future, what's going on for the future. We also invited all these world leaders, scientists, investment management to go together to talk about, what technology and science going to be for the future. And we're going to listen to many sharp talks. All these great figures was from each field. They will share the sharp ideas, the latest results. So it's not only a platform for all these big names, for these shining stars. We also invited you to share your stories. So it's time for you to deliver a talk here. So you are welcome to submit your materials online. So we have many sub-sessions, parallel sessions. You are welcome to submit your materials, and we're going to pick up someone to share that. And it's our time also invite you to be superstars on the stage. If you are college students, welcome to apply for Innovation Award. If you are young scientists, welcome to apply Young Scientist Award. If you already have your startup, welcome to our Startup Award. All these applications is due April 1st, so you have about months to submit it. And it's time for us to go together. We have all these people here who want to work together with you. So we from different continentals, different countries, different areas. So welcome to join us. And it's our time to go to Davos. Now the pre-registration is open now. You are welcome to submit online and go summit together.
在持续发展的社会中，电子元器件需要应对日益复杂化的要求和越来越高的期待。技术革新浪潮汹涌而至，对其高性能的需求、无限小型化的追求，成为行业发展的必然趋势。多层陶瓷电容器、电感器、高可靠性元器件、太阳诱电支撑起全流程的先进技术，提供给用户全方位的服务。为了科技的发展，我们一代代太阳诱电人肩负使命，激情拼搏，以极致的匠心独运，呈现出史诗级作品。研发历程感动了无数电子工程师。太阳诱电将